Bio 47, we're going through Lab 17, the urinary system. What we'll be going through is describing the basic anatomy and function of the urinary system. We'll be tracing blood flow through the kidney. We'll be looking at the location of the urinary system in the abdominal cavity. We will be understanding the internal kidney anatomy and identifying the microscopic structures, so looking at some histology as well. And I'll generally be following the pages in your lab manual. So the first part is to check out some of the gross anatomy of the urinary system. So we'll be looking at the location of the kidney first. The kidneys are located against the dorsal abdominal body wall. They're lateral to the vertebral column at about the level of the 11th and 12th ribs. They're held in place by the overlying parietal peritoneum, the surrounding organs, and some connective tissues. So starting at the visceral layer, the entire kidney is surrounded by a fibrous capsule, and that helps to maintain the shape of the kidney and providing physical support. There's some adipose tissue that covers over the fibrous capsule, and then there's the renal fascia, which is continuous with the parietal peritoneum. The kidneys are all behind the parietal peritoneum, a position that's described as retroperitoneal. And the gross structure of the kidney is that the kidney is convex on its lateral border and concave along its medial border. There's an indentation that's called the hilus along the medial border, and that's the entry point for the renal artery, shown in red here, exit for the renal vein, shown in blue, and for the ureter, which is shown in yellow. If we look at the sectioned kidney, this is a frontal section through the kidney, then you can see some of the internal gross structures. So here would be the hilus, this is the medial side, and this would be the lateral side of the kidney. And the hilus opens into this internal cavity, this open space called the renal sinus. And it has the draining structures of the kidney. It has nerves, it has blood vessels, and it's packed with adipose tissue. That's what's shown in yellow here, is some of this adipose tissue that packs the renal sinus. The functional kidney tissue is divided out into two layers. The outer layer is the cortex, and the inner layer is the medulla. In the medulla, we find somewhere between 6 and 18 conical shaped structures. Those are the renal pyramids that end in the renal papillae, and separating them is supporting tissue. That's the renal columns, essentially the same as the cortical tissue. Urine is produced within each pyramid and drains through the apex, through this papilla, into a minor calyx. The minor calyces merge and drain into major calyces, and in turn that all drains into the renal pelvis, which is continuous with the ureter. And here's a cadaver view showing that posterior dorsal body wall there. There's showing the right kidney and the left kidney. On top of each one of them would be the adrenal glands or suprarenal glands. Remember that those serve to release adrenaline or epinephrine. And then draining down from each kidney, more clearly visible here, is the ureter, that tube that's leading to the urinary bladder. So the ureter, simply an extension of the renal pelvis. It's also retroperitoneal. It's smooth muscle that's lined with transitional epithelium. There's one that comes down from each kidney and drains urine into the urinary bladder, which is on the floor of the pelvic cavity. The urinary bladder is a hollow, smooth muscle organ. It functions to temporarily store urine. It's located just inferior to the pelvic cavity in a retroperitoneal position. The inner lining of the bladder is folded into rugae, similar to the stomach, which allow the bladder a greater ability to expand to up to 1 liter or more. So a triangular region right here. And it's a region in the lining of the urinary bladder. It lacks rugae and it has a smooth surface. This is called the trigone. So this trigone is formed by these drainage points for the 2 ureters and the opening to the urethra. And this serves to effectively drain urine from the bladder into the urethra when the bladder contracts. The bladder contracts when this 3 layers of smooth muscle, collectively referred to as the detrusor muscle, causes compression and an emptying of the bladder. Then the urethra is that single tube that leads from the bladder to the exterior of the body. Its route and its function are different between males and females. In the female, it's solely for the transport of urine, and it's shorter. In males, the urethra drains not just urine, but also semen, and it's longer. It has a longer route out of the body. The exit from the bladder in both is guarded by the inner urethral sphincter, a ring of smooth muscle, and by the external urethral sphincter, and that skeletal muscle that's consciously controlled. And your lab manual gets back into the kidney. So now we're observing a kidney dissection. On the kidney, you can observe the renal hilus, where the ureter is draining out. This one does not show the renal artery and the renal vein, but they would be coming in at the hilus also. You can see this tissue, that outer layer, that's the cortex. And then this right here would be the renal medulla. It's a darker middle region. You can see the renal pyramids that drain into the minor calyces, and the tissue between them is the renal columns. The renal sinus is that internal kidney space. It's filled with renal structures, the blood vessels, the nerves, that proximal portion of the ureter, and the renal pelvis. You can see a minor calyx and a major calyx where they've merged together, and then draining into the renal pelvis continuous with the ureter. Then your lab manual describes the blood flow through the kidney, something that we talked about in lecture. The first one, where blood is entering, that's the renal artery. The renal artery is carrying blood that's filled with waste products from the body to be filtered in the kidney. So the renal artery enters into the renal hilus through the sinus, and then it progressively branches into smaller vessels as it's traveling from the hilus to the cortex. The first branch would be the segmental arteries. They pass through the renal sinus, and the interlobar arteries pass between the renal pyramids through the renal columns. The arcuate arteries are branches off of the interlobar arteries and pass over the renal pyramids. And then you have interlobular arteries that project up into the renal cortex. In your lab manual, they're called cortical radiate arteries, but we're going to call them interlobular arteries. From there, if we enhance over here, we have, we're looking at a section here. This would be the arcuate artery, this would be the interlobular artery, and then you can see the afferent arterioles. These would be supplying individual glomeruli. Not shown here is the glomerulus, that fenestrated capillary network that is closely associated with the glomerular capsule. Blood is carried away by the efferent arterioles. Those drain the glomerulus. and they supply blood to the next step, the paratubular capillaries, those capillaries that surround the nephron and absorb reclaimed blood constituents. So we have a capillary glomerulus to capillary paratubular capillary system. That's a portal system, and allows for exchange between the urinary and the cardiovascular system at both of these points. From there, blood drains into interlobular veins, and then into arcuate veins, into interlobar veins, and then into the renal vein, which would be this. So in anatomy, it's typical that oxygenated blood is shown in red. So that's what I've indicated in the text here. And deoxygenated blood is indicated in blue. So that's what's shown over here is oxygenated blood coming in, deoxygenated blood leaving, and blood that's partially oxygenated is interacting with the capillaries. So it's at the capillaries where oxygen is picked up by cells. And here we're getting a view of some of these nephrons, the functional units of the urinary system. And this is where blood is being filtered and being modified. And this image shows the two types of nephrons, the cortical nephron and the juxtamedullary nephron. Cortical nephrons are 85% of nephrons. They're responsible for producing the majority of urine. And then the juxtamedullary nephrons, those that are closer to the edge of the cortex and the medulla, those are going to be responsible for concentrating urine. So the first part that we're going to look at is the renal corpuscle. The renal corpuscle is the glomerulus, that ball of capillaries that's enclosed by the expanded region of the nephron, the glomerular capsule. So the whole thing is the renal corpuscle, and then it's comprised of the glomerulus and the glomerular capsule, sometimes called Bowman's capsule. The glomerulus is the capillaries that are acting as a filter. So they have holes in them called fenestrations, and they allow blood constituents to be separated by size. So larger constituents, things like blood cells and proteins, are retained by the glomerulus and get pushed out into the efferent arteriole, but smaller things like nutrients and minerals are filtered out into the glomerular capsule. And that filtered fluid, which is now called filtrate, gets directed into the tubular portion of the nephron, where we can have active reabsorption of some of those valuable constituents of the blood. So the glomerular capsule, Bowman's capsule, is that expanded region of the nephron that surrounds the glomerulus, and it's composed of two layers of cells. There's the visceral epithelium, which are the really highly modified cells that cover the surface of the glomerular capillaries. These cells function to further enhance the filtering process. They form small finger-like extensions called pedicles and filtration slits over the surface of the blood vessels. These cells are called podocytes, which means foot-like extensions. So podocytes are the cells that make up the visceral layer of the glomerular capsule. Outer layer is the parietal layer of the glomerular capsule. So the parietal layer of the glomerular capsule. This is simple squamous epithelium that forms the expanded region of the capsule. And from there, from the capsule, we have filtrate that flows through the renal tubule. And the renal tubule is composed of highly specialized cuboidal cells that function to modify the blood filtrate as it passes through the tubule. And this extensive processing results in the production of urine at its completion. The first part is called the proximal convoluted tubule. It's the first portion of the nephron, and it It's a highly twisted region that receives and modifies the filtrate, and it functions to reabsorb a majority of the valuable nutrients and salt and water that were initially filtered into the glomerular capsule, and these get reabsorbed into the peritubular capillaries to go back into general circulation. Then there is a portion that drops down called the loop of Henle, or the nephron loop. This is a very thin region of the nephron that forms this hairpin drop down into the medulla. And it uses countercurrent exchange to concentrate urine, something that you'll discuss a lot more in physio. After that is the distal convoluted tubule. That's the last segment of the nephron. It's distant to the renal corpuscle, that's why it's distal. It's responsible for secretion of additional waste into the filtrate, as well as some reabsorption. And the distal convoluted tubule drains what's now urine into the collecting duct. The collecting duct are those microscopic tubules that collect the newly formed urine, and they're going to drain into the renal papillae, into the minor calyx, into the major calyx, and into the renal pelvis. So now we'll do a little histology. And here we're looking at some histology of kidney tissue. And remember I told you that you'd be able to see the renal corpuscle because it'll appear spherical with this sort of halo around it. So you can tell that you're at the border of the cortex, so this would be the outer side, and the medulla, because all of the corpuscles are located in the cortex. So this would be, these over here would be those juxtamedullary nephrons, and those up here, these would be cortical nephrons. So it's depending on where the corpuscle is located. Is it close to the medulla, or is it very distinctly up in the cortex? So you'll be able to see the renal corpuscles within the cortical region of the kidney, and you'll be able to see the glomerulus, that tuft of capillaries, as well as the renal capsule, that simple squamous epithelium that surrounds the glomerulus, and you'll be able to see this halo, the capsular space. You'll also be able to see sometimes cross sections through renal tubules. So you'll be able to see distal and proximal convoluted tubules. You don't need to be able to distinguish them. So you can see that they are lined with that simple cuboidal epithelial tissue. And then down in the medulla, you'll see some that are larger, and those are the collecting ducts that have urine that's been collected from multiple distal convoluted tubules. So here's another look, and here we're just looking at cortex because we're seeing a lot of glomeruli. So there's one, there's one, there's one, and there's no distinct border where they stop occurring. So we're just in the cortex of the kidney right now. You can also see sections through some of the convoluted tubules. And again, you don't need to be able to distinguish between proximal and convoluted tubules. If we get a little bit closer, then now we're under 300 times. And you can see, again, in the middle, the glomerulus, that tuft of capillaries. And that's everything in here. You won't be able to distinguish the podocytes, that visceral layer of the capsule. You'll see the capsular space, that halo around the glomerulus. And then you'll be able to see the simple squamous lining of the renal capsule. And you'll be able to see some of these simple cuboidal cells that are lining either the proximal convoluted or distal convoluted tubule. And again, you don't need to be able to tell those apart. So make sure that you go through your lab manual pages, do the review on pages 11 through 15 of lab 17. I've also set up some visible body activities for you to do. And please let me know if you have any questions about this lab as well.